Thank you, Dan. Good morning, everyone. My apologies for not being here in person, but I did want to be able to at least share with you some of the research that we've been conducting on the use of parasitoids to control brown marmorated stink bug. We've been talking about this invasive insect for the last 7 years, and we think that we have a potential solution, at least in part, to reduce the populations of BMSB in New York State. So with that, I'd like to introduce you to some aspects of the project. The first being the rationale behind management of brown marmorated stink bug. In many respects, it goes without saying that an insect that causes this level of injury needs to be managed. The difficulty that we've had is the unpredictable nature of the insect in the field. We can set traps and we can monitor the presence of brown marmorated stink bug, but they don't always correlate to the insect being in tree fruit. And so if we can lower the population of BMSB, then the threat, presence, damage that they produce could be reduced significantly. So with that, we've taken on many different trials. This one, we believe, has the greatest potential for actually bringing the population into a much more reasonable presence and management program. The Trissolcus Japonicus is in a group of wasps known as Scelionidae. They're very, very small. You can see that relative to the size of a pencil point, it's about half the size of the carbon tip, 2 millimeters in length. The female samurai wasp will lay its own eggs into the eggs of brown marmorated stink bug. The wasp that is developing within the egg then feeds on the BMSB nymph, and after the wasp has pupated, it then takes its mandibles and chews its way out of the top of the egg, emerging as an adult wasp to begin the cycle again. So in this diagram on the left is the life cycle of brown marmorated stink bug. In the spring, it emerges as an adult from overwintering sites, whether that be in the woodlands or in urban settings, people's homes, outbuildings. It flies into either tree fruit or woodland trees to lay its eggs. Those eggs then hatch. The nymphs emerge. They go through 5 different instars. The nymph can feed on a number of different crops. They've been found on over 200 different hosts throughout the United States. Once the nymphs transition and emerge as adults, the adults can then fly off to feed on either tree fruit or other vegetable crops, or back into deciduous tree hosts. The intersection between the samurai wasp and the adult is that point at which the adults begin to lay eggs. The samurai wasp search for egg masses, and it finds those by sensing stink bug footprints. These are volatiles and other chemicals that the stink bug leaves behind. So once the wasp finds the eggs, it determines whether or not the eggs are viable. Begins to oviposit in them. It'll then guard the eggs, and as the eggs develop, they begin to darken. The wasps then emerge as adults. The males usually emerge first and wait for the females to emerge. They then mate and begin the process over again. Most of this, if not all of it, will occur in deciduous forests along clusters of trees such as locust and ailanthus, and it will allow for the wasp a high rate of survival because it's not going to be in the crop. So to give you some background on Trissolcus japonicus, Kim Holmner is a researcher from the USDA ARS in Newark, Delaware. He works as director of the Beneficial Insects Introduction Research Facility. In 2007, after BMSB got established, he surveyed natural enemies of BMSB in Asia. He then returned with live parasitoid specimens. He held them in quarantine facilities, and he began to study the effects of BMSB on the pentatomid population. He found that the samurai wasp was highly successful as a parasitoid, and it can reduce the population of BMSB as high as 80%, which has been found in China in recent studies. In choice and non-choice tests, the parasitoid wasp has been found to be highly effective. It can parasitize 60 to 100% of those eggs in a BMSB cluster. So not only are they highly effective, they're very specific. They choose BMSB over other pentatomid eggs. However, if they're not given a choice, then it has been found that Japonicus can oviposit into the eggs of a predatory stink bug species known as the spine soldier bug, Pedesus maculaventis. In 2014, the advent of populations of Trissolcus were found in the wild. In 2015, they were found in Washington State, as well as in Washington, D.C. and in Winchester, Virginia. And in 2016, it spread throughout West Virginia, Maryland, Delaware, New Jersey, and that's when we found it in Marlboro, New York. And they also saw it in Washington and in Oregon. So the population is quite diverse in terms of its location. You could see at this point it's been found in the east and in the west coast. It was recently found in Pennsylvania. From 2017, the Oregon group of scientists and those of us at the Hudson Valley Research Lab began to develop colonies and began spreading Trissolcus throughout the state. What has been found with the wild or adventive populations is really unique. There are actually 3 distinctly different genetic population groupings. That work has been done by a woman by the name of Marie Claude Bon. She works at the USDA Ag Research Station at the European Biological Control Lab in Montpellier, France. So we, along with many other researchers, have been sending our finds of these adventive Trissolcus japonicus populations to France, where she then extracts these microsatellite gene markers and is able to differentiate these populations into 3 major groupings. So you can see the Marlboro, New York population in the lower left-hand corner, distinctly different from Vancouver or Portland and different from Winchester, Virginia, with regards to these DNA markers. So the most important find here is that these adventive finds, these wild populations, are not related at all to the populations that were held in quarantine. And so these are independent and unique introductions of Japonicus. So once we located these in New York, we needed to come up with a mechanism, a way of distributing them throughout the state. The first step was to obtain a Liberation of Wildlife permit, which we submitted to the DEC. And they reviewed this and basically concluded that Trissolcus japonicus does not require a license or permit from the DEC to begin the movement and the release of the wasp throughout the state. And that's because although it's invasive, it's not considered a threat, nor is it an endangered species. And so with that, we were able to begin the process of moving the wasp and expanding its range in the state. So over the last 8 years, we've been monitoring brown marmorated stink bug. We've seen the increase in the urban and agricultural areas since 2010. We established a colony of BMSB with which we conduct bioassay studies. And then more recently, we've been employing the eggs of brown marmorated stink bug, which are laid on jalapeno pepper leaves. So we have these plants in the lab that they feed on and they lay their eggs on, and we use those as sentinel eggs. We believe that their movement throughout the pepper plants leaves behind more chemicals and possibly are more attractive to parasitoid wasps. So we began the survey of the native and adventive parasitoid complex using these sentinel eggs to attract Trissolcus japonicus in New York. But first we freeze them to minus 80 degrees Celsius for 4 minutes to kill off all the BMSB nymphs that are developing inside those eggs. We then developed a Trissolcus japonicus colony, and we began establishing release sites. So in contact with Art Agnello, Tessa Grasowitz, and Debbie Breath, they acted as cooperators on Western New York, and they're the ones that located the sites, placed the eggs out, and began monitoring for both native parasitoids, Trissolcus japonicus, and later helped us in establishing release sites in 2017. So these sentinel egg surveys that we have been conducting for native parasitoids began in July. We had 7 sentinel sites, 2 in eastern New York, that were selected to survey for parasitoids. The eggs were fixed onto known BMSB hosts. We used 7 different host plants. The majority of the plants were locust and sugar maple we were using, as well as the Ailanthus altissima, or the tree of heaven. But we also used wild grape and a number of different host plants that BMSB lay their eggs on. These eggs are sent overnight to cooperators. We place them in Wayne, Orleans, Ontario, Columbia, Ulster, and Dutchess counties. We then, after they were placed, recollected them on a 5 to 7 day schedule and then put them in our environmental chamber at 25 degrees Celsius, 14 hours of daylight. We then monitored the emergence of the parasitoid, and then all of the emerging wasps we sent to Elijah Ptolemus, who is now at the University of Florida in Gainesville. The adult parasitoids that we reared from Sentinel eggs were then provided 90% honey water solution. We used 1 microliter, very small droplets, on the dish for the wasp to survive and reproduce. And this is what the Sentinel eggs look like when they're stapled onto foliage. So these Sentinel eggs were placed on 10 farms, 3 to 24 clusters per site per week, for a total of about 2,700 Sentinel eggs. It's about 80 plus egg masses over the course of that period of time. So what we found was 3 different species. We found 2 native Trissolcus euschistii and Telenomus pedicei beginning in late June. Relatively few emerged from those BMSB eggs. And in Marlborough again, which was the only site we were able to capture Trissolcus japonicus, we had emergence beginning on the 7th of July, in which 96 adults emerged. And it was from that population in 2017 that we began our colony, and we began parasitizing eggs for redistribution of the wasp. We then reared the wasp on frozen BMSB eggs. The first parasitized eggs were sent to cooperators beginning on the 15th of September. So it took us quite a while to establish and develop the colony, and we didn't begin until kind of late into the season. So we weren't sure that this was going to be effective. The parasitized eggs were put out on 32 different farm sites. It encompassed 25 farms in 5 counties throughout the state. The placement was at about 5 or 6 feet in a number of different tree hosts that you can see here. Most of the sites were placed adjacent to orchards where stink bug injury had occurred. So the placement of parasitized eggs, 24 farms, 6 counties, 32 sites, about 2,300 eggs on 7 different host plants is what we're looking at here. Beginning the 15th of September, and the last placement was on the 6th of October. This is a visual mapping of the Sentinel egg placement sites in the large white circle and the samurai wasp release sites in the smaller yellow circle. When we recollected the eggs, what we found was that we were able to get to 11 of our 32 sites beginning in late October to November. We lost a lot of our eggs because of defoliation in the fall. In 11 of the sites, 77% of those clusters were recovered. In 3 of the 11 sites, we were really happy to see samurai wasps guarding the egg clusters. So we know they emerged. We know that they were providing behavior that was very typical of this wasp, and that it was likely to be present in the field. We also found 168, or 24.4%, of the adults actually emerged from those eggs. So we had 719 successfully emerged adults. I was very happy with that result. Less than 1% partially emerged from the egg, and about 66% of the eggs showed no sign of emergence. Now, we think that may have been due to cooler weather that was occurring later on in September into October. So the eggs were parasitized, but they were unsuccessful in their development. Now, as a follow-up to all of this, we put out Sentinel eggs in 2 of our sites where we released the samurai wasp. And we did that in order to try to recapture the samurai wasp in these eggs. And so we placed Sentinel eggs about 30 meters from where Japonicus was released in 2 of these sites. What we found was that Japonicus did in fact parasitize these eggs from 2 of the sites between the 15th of September to the 3rd of October. So those were in Monroe and Orleans County. So we feel confident that that establishment did occur in those 2 sites, and that Trissolcus now has a chance to get through the winter and be present in the spring. Now this winter's been pretty tough. We've had some low temperatures, and Trissolcus japonicus, just like BMSB, has a low temperature tolerance of about 0 degrees Fahrenheit. Once it drops much below 0 degrees, then we see over 90% mortality in BMSB, and we think Trissolcus japonicus has a similar tolerance to that. So we'll hopefully find out this year whether or not they've overwintered successfully. And we're going to do that by placing, again, sentinel eggs in these release sites. We're looking for Trissolcus japonicus as well as other natives. We're going to provide them 5 days of field exposure, return them back to the lab, see if we have Japonicus successfully rear, which would confirm establishment. However, if we don't see them emerging, we're going to reestablish these sites earlier this year. We hope to establish an additional 200 sites. So we can send Trissolcus japonicus eggs to New York growers upon request and help you with the process of returning those eggs and determining whether or not you've had emergence. So with that, I leave you my email address, and I'm hoping that you would, if you're interested in the project, if you're interested in obtaining these parasitoid eggs, that you contact us, and we will overnight the eggs to you for you to put out, and then provide you with a return envelope so you can send them back to us in a week's time and help us to be able to determine whether or not those wasps emerged from your site. So with that, I'd like to thank you all for your attention. I'd also like to thank our lab staff for their support from Ag and Markets, ARDP, the Dow AgriScience and Bayer, the New York State Specialty Crop Research Initiative, as well as, and I can't state this enough, the New York orchards, farmers, managers that have contributed to the Hudson Valley Research Lab to be able to maintain the facility and run the facility as it should be. So thank you for all of your generous support. See you soon.